Hello everyone, welcome to the second part of immune system disorders in Robbins. So in last class we were discussing about the basic introduction to disorders of immune system, and we have ended that session by mentioning few words about primary and secondary lymphoid organs. You know, bone marrow and thymus both are the primary lymphoid organs, whereas lymph node, spleen, cutaneous and mucosal lymphoid systems, all these are the secondary lymphoid organs. So let us start. What are lymph nodes? I'm not going to discuss elaborately, fine, because in depth we will see the structure, okay, the various regions of the lymph nodes in our hematology. Okay, in WBC disorders we have to discuss this again, fine. So now let us see about lymph nodes. What are they? They are simply the aggregates of lymphoid tissues, fine. Yes, here only maturation of T lymphocytes. The T lymphocytes what will fight with antigen encounters, everything takes place in the lymph nodes. Lymph nodes have some of the different areas of there in lymph nodes, fine. As far as lymph node is concerned, it has an outer cortex and an inner medulla. It has an outer cortex and an inner medulla. Remember, inner medulla has mainly B lymphocytes. Outer cortex, okay, also have lymphoid follicles which contain follicular dendritic cells. I have told you what are follicular dendritic cells. And of course the cortex also have what we call it as the B cells. B cells are found in cortex also. There is a region between the cortex and medulla, that is what we call it as paracortex. So you can see this is the medulla, cortex, follicles, and this is the region. Okay, this is the region I am drawing here. This region is what we call it as the paracortex. In paracortex we have the T lymphocytes. T lymphocytes are present in the paracortex. Okay, I hope that is clear. Fine. Then we have spleen. Okay, fine. So this is, you can see, this is again lymph node. You can see the T cell zones. Fine. The dendritic cells, they are follicular dendritic cells. They are present in the cortex again. Okay, the naive T cells. What do you mean by naive T cells? Naive means, yes, the B cells or the T cell, they have not encountered with any antigen. That is the meaning of naive T cells. Fine. Then we have spleen. It is an abdominal organ. It is usually called as the graveyard of RBC. Okay. Again, it will prevent the antigens from getting into the blood, everything. Okay. It will degrade all the antigens, everything. Fine. Yes. Next we have cutaneous and mucosal lymphoid system, that is the MALT, mucosa associated lymphoid tissue, bronchial associated lymphoid tissue, and the gut associated lymphoid tissue. The best examples we have, they are nothing else but called as the pharyngeal tonsils and the Peyer's patches of the intestine. Fine. Yes. Then I have told you the lymph nodes, the B cells are present in the follicles, okay, and in paracortex I told we have the T lymphocytes. As far as spleen is concerned, we have white pulp and the red pulp. Red pulp is mainly having the splenic sinusoids, whereas in the white pulp only we have the B lymphocytes. So remember, the B cells are mainly found in white pulp, whereas T cells are found in something called as PALS. What is PALS? PALS stands for periarteriolar lymphatic sheath. Periarteriolar lymphatic sheath. Okay, yes. So once that is done, we have to know what do you mean by lymphocyte recirculation. So the lymphocytes, they will constantly recirculate between the tissues and home to particular sites. For example, the naive lymphocytes, so actually these naive lymphocytes, they are mainly present in the secondary lymphoid organ. They will migrate to the areas where there is any inflammation. So there will be always all the lymphocytes, they won't always sit in the same place. They will be always on circulation. Fine. So all these are very basic things, not that much importance as far as path is concerned, but coming to a very, very important concept, that is called as MHC. What is MHC? MHC stands for major histocompatibility complex molecule. Okay, MHC stands for major histocompatibility complex. So in humans, so what is the role of this MHC molecule? Antigen recognition by T lymphocytes. Am I right? Yes. And remember, this MHC complex has very important role in autoimmune diseases that we will discuss in the coming classes. So remember, in MHC molecules, okay, I'm going to say just the very important salient features. The MHC molecules, they are called as human leukocyte antigen because they are mainly first discovered on the human leukocyte. They are mainly located on chromosome number 6. Very important point. It is highly polymorphic. That is, it is made up of there are lot of thousands of alleles are present. Alleles are present for this MHC. Fine. And remember, other than identical twins, definitely each of us have different MHC. Fine. Like, okay. Then we have different classes of MHC, class 1, class 2 and class 3. As far as path is concerned, we have to study in detail about class 1 and class 2 MHC molecule. Class 3 MHC molecules, which include some cytokines and other proteins, which will study in detail in immunology and microbiology. So now let us see class 1 MHC molecule. As far as class 1 MHC molecule is concerned, what is the important point? Number 1, the location. Class 1 MHC molecules, they are expressed on all nucleated cells and platelets. All nucleated cells and platelets. Fine? Yes. So actually, what all things it is having? It is actually a heterodimer. It is a polymorphic. Heterodimer means what? It is having what? Yes, two different types of chains. One is the alpha chains, which are alpha 1, alpha 2 and alpha 3 in number. And finally, the second different set of chain is called as the beta 2 microglobulin. That is why it is called as a heterodimer. Why it is polymorphic? Yes, alpha 1, alpha 2, alpha 3, beta 2. All these are completely different entity. That is why polymorphic. And remember, this alpha chains of the class 1 MHC molecule, they are coded by 3 genes. They are called as HLA-A, HLA-B, HLA-C. You can see in this picture. Okay, HLA-A, B and C, they are class 1 MHC molecule, and you can see very beautifully in this picture, it is having alpha 1, alpha 2, alpha 3, beta 2 microglobulin. All these are completely different in their arrangement, in their structure. That is why we call them as polymorphic. And why we call it as heterodimer? Because we have both alpha chain as well as the beta chain, heavy chain as well as light chain. Fine? Yes. And as far as class 1 MHC molecule is concerned, I told you this MHC molecules helps in antigen recognition. Am I right? Yes. And they binds with the T lymphocytes. So you should know at what site, okay, the antigen will bind and at what site the T lymphocyte will bind. So these are very, very important. These are repeatedly asked in all the PG entrance exam question, all the central institute exam like INI CET. Remember, as far as class 1 MHC molecule is concerned, the peptide binding cleft or the antigen binding cleft is located between alpha 1 and alpha 2. Okay. Whereas the T lymphocyte, I told you what is the rule? 1 into 8 is equal to 8. That means MHC1 will bind always with the CD8. So the CD8 will bind with the alpha 3. Okay, CD8 plus will bind with the alpha 3 gene. So the peptide binding cleft, that is the antigen binding cleft, is present between alpha 1 and alpha 2. Whereas the T lymphocyte, so we have seen where the antigen will come and bind. The antigen will come and bind between alpha 1 and alpha 2. Whereas the T lymphocyte, that is CD8 in case of class 1 MHC, it will bind with that of the alpha 3. Okay. That is about class 1 MHC. Fine. And remember, this class 1 MHC molecules, they are targeted against virus and tumor specific antigen. Whenever there is a viral infection or whenever there is a neoplasm, what will happen? What will happen? Yes, it is this CD8 or cytotoxic T cells that will come into action and they are going to kill these cells. Fine. Viral infected cells and tumor infected cells. Okay. Sorry, tumor cells. Fine. Okay. Once that is done, I told you CD8 plus. Okay, that is the most important point. Once that is done, let us see about class 2 MHC molecule. Remember, class 2 MHC molecules, where they are present. Okay, remember, they are mainly expressed on antigen presenting cells like macrophages, B lymphocytes and dendritic cells. What about class 1 MHC molecules? They are present in all nucleated cells and platelets. And class 2, what is the formula here? Class 2 into 4 is equal to 8. That means, of course, it will bind with the CD4 T helper cells. Let us see the structure of class 2 MHC molecule. See, as far as class 2 MHC molecule is concerned, it has different region. They are called as a DP. Okay, they are DP, DQ and DR. Here we have A, B and C gene. Here we have DP, DQ and DR genes. Fine? Yes. And what are the structures? Again, it is heterodimer and it is polymorphic. Why it is heterodimer? Because it is having both alpha chain and beta chain. And why it is polymorphic? Here we are having alpha 1, alpha 2, beta 1, beta 2. There in class 1 we have alpha 1, alpha 2, alpha 3. Okay, here we have alpha 1, alpha 2, beta 1, beta 2. Here again 2 points. At which side the peptide is going to bind and which side the CD4 helper cells are going to bind. Remember, the CD4 plus helper T lymphocytes, CD4 cells, they are going to bind at beta 2 region. Whereas the peptide binding cleft or the antigen binding site is between alpha 1 and beta 1. There it is between alpha 1 and alpha 2. CD8 is with the alpha 3. Okay, here it is between alpha 1 and beta 1. The peptide binding cleft is present and beta 2 binds with the CD4 plus cells. Okay, these are very basic points. Okay, these points you cannot miss. Okay, of course, you are not going to get any questions regarding MHC because it is a question purely from immunology and microbiology. But definitely you should know what these are some very basic concepts. And I have already told you they exhibit polymorphism. Other than identical twins, no two individuals have same MHC molecules. Fine? Yes. Okay. What are the important roles? Remember, this MHC cells, MHC molecule, they mainly play a role in what we call as the T cell mediated immune response. They almost have, okay, I can't say they have no, it doesn't mean that they don't have any role in humoral immunity. No, they have predominantly more role in cell mediated immune response. Fine? Yes. And what is the major reason why MHC molecules? Because, you know, each MHC molecule will respond to different classes of antigen. Why? Because each of these MHC molecules have different peptide binding site. Okay, that is one of the reasons for this polymorphism. I told you each MHC molecule, we have thousands of alleles. Am I right? For this MHC molecule. So each MHC molecule have different, for example, I am telling MHC1. I told you, MHC1 mainly they respond to virus infected cell as well as tumor cells. So they are mainly specific for that MHC1. At the same time, MHC2, they are mainly for bacterial or other microbial infection like fungus, everything. MHC2 will come into action. So microbial fungal infection, MHC2 will come. So each of the MHC molecule is designed in such a way that they are highly antigen specific. They are highly protein specific. Fine? Yes. Okay. Once that is done, let us see some of the important cytokines. This is one of the most favorite area of question for the Central Institute exam, like INI-CET. Okay, they will get multiple questions from this. What are the cytokines which are produced from the innate immune response as a result of adaptive immune response? And finally, what are the factors which stimulate hematopoiesis? First, let us see in innate immune response what are the what cytokines there. Please, there is no other way how to simply mug it up. TNF, okay, interleukin 1, 12, type 1 interferons and gamma interferons. These are the cytokines which are produced as a result of innate immune response. Whereas in case of adaptive immune response, see again we have interferon gamma. Interferon gamma is common to both of these response. Fine. And remember TGF beta, interleukin 10, interleukin 2, 4, 5 and 17. Okay, all these are cytokines are produced as a result of what? Adaptive immune response. Then what are the factors which stimulate hematopoiesis? We call it as CSF. What are they? Colony stimulating factors. I think you would have already studied about this in physiology. We have two types, mainly granulocyte, monocyte colony stimulating factors. Okay, and interleukin 3 and interleukin 3. So these and all you have to just mug it up. Fine. Okay. So I told you. So far, how for suppose a pathogen has entered in your body. We have already discussed about what? Yes, pathogen associated molecular pattern, PAM, DAMP, everything. So we have to first recognize the antigen. Once that antigen is recognized, it has to be what? Taken out by the antigen presenting cell. It will present via the MHC through the T lymphocyte. Finally, the T lymphocyte will initiate whether it is a CD8 or CD4, it will initiate the cell mediated immune response. Finally, killing by cytotoxic cells, everything happens. Fine? Yes. So that is the major. Okay, recognition of antigen is one of the very important phenomenon. What in our immune response. Fine. So remember, even before these antigens are recognized by our T and B lymphocyte, the microbe will elicit an immune response. How? I told you, via the toll-like receptors, pattern recognition receptors. Actually, that is the first line of defense. That only first will comes into action. Fine? Yes. So how that will happen? So normally I told that what is the process? Antigen will come, recognition, APCs, antigen presenting cells, MHC, T lymphocyte, cell mediated immune response or humoral response. That is what is going to happen. But before that, what happens? Before that, what will happen? Yes, there will be recognition of antigens by pattern recognition receptors. And these pattern recognition receptors are mainly a part of what? Innate immune response or adaptive immune response? Of course, it is innate immune response. And during this innate immune response, fine, this microbe will also activate the antigen presenting cells, but not like the conventional way, like to the T or B lymphocytes. Okay, we have some co-stimulators. Co-stimulators are nothing else but some of the molecules, okay, which are activated as a result of innate immune response, okay, without any MHC activation. And there is no MHC activation here. In innate immune immune response, we don't have any MHC complexes. Only in adaptive immune response, we have MHC, all those steps. Here, via some two important co-stimulators, what will happen? The T lymphocytes are activated. So it is actually an indirect activation. Usually for activating T lymphocyte, what is the pathway? Via the MHC. But here there is no MHC. Okay, because the innate immune response is going to directly activate the T lymphocyte via co-stimulator molecule. The two most important co-stimulator molecules are CD80, CD86. It is a JIPMER previous year entrance exam question. Okay, it is CD80 and CD86 are the two important co-stimulator molecule, and these are recognized by the CD28 receptors on the T cells. For that, the T cells will be activated. Here there is no role of MHC. No role of MHC. Fine. I hope it is very clear for you. Yes. Fine. Then now let us see. How the T lymphocytes are activating and how there is an elimination of intracellular microbes. Fine, let us see that through this picture. See, we have CD4 8 cells, CD8 cells, finally T cell proliferation, differentiation, okay, finally phagocytosis, everything, cytotoxic T cells, they will cause killing, inflammation, okay, all those will happen. These are all usual pathways, okay, nothing special to mention. And we have different types of T helper cells. This is a very, very important pick, okay. T helper 1 cells are there, T helper 2 cells are there, T helper 17 cells are there. This entire thing you have to study. T helper 1 cells mainly produce interferon gamma. This interferon gamma will further produce much more interferon gamma and interleukin 12. And they causes macrophage activation, killing of intracellular microbes. They are mainly playing a role in autoimmune disease. Whereas T helper 2 cells, rule of 2. Okay, rule of 2. T helper 2 cells produce interleukin 4, mainly interleukin 4. They causes IgE production, so parasite allergic response. Okay, we will discuss again this in type 1 hypersensitivity reaction. Type 1 hypersensitivity reaction, which is an allergy mediated reaction. In the T helper 17 produce interleukin 17, TGF beta, monocytes, neutrophils recruitment. They mainly act against extracellular bacteria. See, that is a major difference. TH1, T helper 1 cells mainly act against intracellular microbes, whereas T helper 17 is extracellular microbes. Fine. Yes, of course, they play a role in autoimmune disorders also. Please go through this table. One question definitely. Which of the following cytokines are produced by T helper 1 cell or 2 cell, T helper 17 cell? Remember, T helper 1 means interferon gamma, interleukin 12, macrophage activation, intracellular microbe. T helper 2 means rule of 2, 2 into 2, 4. That means interleukin 4, it is producing IgE, helminthi parasites, allergic reaction. T helper 17 means interleukin 17, they are mainly acting against extracellular bacteria and fungus. Okay. Then we have what? Yes, antibody secretion, class switching, affinity maturation, all these terminology is not that much important as far as path is concerned. Remember, finally they cause neutralization, opsonization, phagocytosis, antibody dependent cytotoxicity, okay, all those. Opsonization is mainly what enhanced by C3b receptors, opsonins, okay, C3b receptors, fine. C3b, C5a, all these are there, okay, yes. Okay, what do you mean by isotope switching and what do you mean by affinity maturations? Okay, yes. Here, I forgot to mention a very important thing. I told you about CD4 helper T cells. Okay, yes. The CD4 helper T cells, okay, they mainly produce, CD4 helper T cells mainly produce interleukin 2. They mainly produce interleukin 2 and that is via what ligand? CD40 ligand. Via CD40 ligand. Remember CD4, CD40. Just one liner, MCQ, one liner. There is no need that you should understand what in depth about the rest of the concepts and all. It's complex. No need for our level. Fine. So remember, we know already interferon gamma causes classical macrophage activation. Already we have studied in inflammation. Fine. Yes. Remember CD4 will activate the cells via which ligand? CD40 ligand. Just remember that. You may get MCQ question for that alone. Okay. Okay, fine. We will discuss, okay, some of the disorders of immune system where there is a deficiency in CD40 ligand, everything. So the protein antigens, by virtue of CD40 ligand, what will happen? They induce the production of antibodies of different classes. Okay, that is called the isotope switching. That means, suppose T lymphocytes are being activated by CD40 ligand as a result of some antigen. What is happened? It is not only going to produce one class of antibody, it will produce different classes of antibody. Initially it will be IgG, from IgG finally it will become IgD, IgA, IgE. Different classes of antibodies are produced as a result of an antigen, single antigen activation. This is what we call it as, yes, this is what we call it as isotype switching. Here comes an important MCQ question. Isotype switching is mediated by CD40 ligand. This question has been asked in JIPMER few years back. Okay, yes. Then as a result, okay, that is one thing. Second thing, these antibodies, these T helper cells will stimulate the production of antibody which are having very high affinities for the antigen. That is what we call it as affinity maturation. What do you mean by affinity maturation? We have different classes of antibodies, right? The T lymphocytes will stimulate only those antibodies which are having very high affinity towards this antigen. So much more in detail about this you can read in what? Immunology. As far as path is concerned, there is no need to know much more. And T helper 2 cells, I am telling, interleukin 4, rule of 2 I am telling, 2 into 2 is 4. And then what? T helper 2 cells mainly help in IgE, allergic reaction. Okay, that is an important point you have to remember. So today in this class we have discussed the very basic, just to know what is the very basic structure in lymph node, spleen, where and all, the B and I told no, this flowchart is important. In cortex we have B cell, paracortex T cells, spleen, white pulp B cells, pals we have the T cells. Once that is done, class 1 MHC, in class 2 MHC. Class 1 MHC, the peptide binding cleft is between alpha 1 and alpha 2, CD8 bind with alpha 3. In class 2 MHC, we have DP, DQ, DR, all those steps. Alpha 1 and beta 1, in between that the peptide will bind, whereas CD4 plus will bind with beta 2. Okay, remember the MHC located on what chromosome? 6. Fine. Then 1 into 8, 8. That means MHC 1 is for CD8, whereas MHC 2 is for CD4. Okay, and in MHC 1 we have HLA, A, B and C. Then what are the important role I have told you. Then class 1 MHC is mainly found in where? All nucleated cells and platelets, whereas class 2 mainly in the dendritic cells. Fine. Then you should know what is co-stimulator, CD80 and CD86. There is no role of MHC there. Pattern recognition receptor, innate immune response, directly activating T lymphocyte via these co-stimulators. Then please go through this. What are the various interleukins and various functions of T helper 1, T helper 2, T helper 17. Remember T helper 1. Okay, how I used to remember? T helper 1 almost looks like I. So I for intracellular. T helper 17 is for extracellular. Fine? Yes. Then affinity maturation and isotope switching. Remember isotope switching is mediated by CD40 ligand. This has been asked in JIPMER. Okay, so this is a very short session. Okay, all these are must know points. You can't skip this point. Just like it's almost like a by hearting. Okay, yes. So please go through these points. Thank you.